Morning, everybody. It's a real honor to be here today. I went to the Darwin Festival, gosh, it was 84 to 89. So, and graduated with a bio degree then. And I know Dr. Case really well. We were just reminiscing a little bit. It's such an important part of the fabric of the biology department and the university, and it's great to be back. So thank you for the invitation. I work for Thermo Fisher Scientific. We're based in Waltham, probably the largest private employer, I think, in Massachusetts and the Commonwealth. We have about 20 facilities here in the US, in Massachusetts, and about 314 worldwide. And we are a life science business looking to do all sorts of things. We helped make the COVID vaccine, for example, for Pfizer with Moderna. We do a lot of work in the life science industry. I'm really honored to be here to introduce Webb Camille. Webb is a 2017 graduate with his BS in biology. Again, same, it's awesome. From Salem State and currently a fourth year MD PhD candidate student at UMass Chan Medical School. He's been involved in multiple extracurricular activities, including serving on the Action for Equity and Inclusion Committee, and of course, Student Government Alliance, which is awesome. I'm a former student government person myself, as well as being a mentor for undergrads through the American Physician Scientists Association. While in medical school, he founded and ran his own nonprofit, One Leaf Corp, focused on food insecurity and climate action in Everett, Massachusetts. He's consistently been an advocate for current Salem State students by attending biology seminar classes, making himself available for networking with current students. And I can say also I have the same passions as a biology grad here with all of you. I've been to, I think, biology seminar a few times in the last year or so. I'm trying to also help get the next generation, into science. So it's a real honor to introduce you, and I'm going to pass it over to Jason for a few remarks for you as well.
Thank you.
Great.
So I just want to say a couple of quick words before I pass it over to Webb and introduce him. For those of you who are here in the room and need to get attendance for one of your faculty members, there will be a QR code on the wall over there during the question and answer session. And I also wanted to just say that Webb is graciously going to hang around with us after his talk and have lunch with anybody that's interested. So please hang around and have lunch with Webb and meet him. So I first met Webb near the end of his sophomore year at Salem State when he approached me about joining my research group to do a research project with me. And I was super impressed with how excited he was about science and how excited he was about medicine. He told me the first day that he walked in that he was going to go to med school. And also I was kind of impressed with his confidence in approaching new Salem State faculty member that he had never met. That was also my first year here at Salem State. And so I decided to give him a project that I thought he could maybe do in a couple of weeks. He got it done super fast, and I was super impressed with the work that he did. He basically repeating an experiment that I had done when I was a postdoc at UMass Medical School, which I hadn't yet done at Salem State. So I quickly advocated for Webb to come spend part of his summer that year at UMass Medical School in George Whitman's lab, which he did, and like rearranged his schedule on the spur of the moment, basically, to come do that. And then he spent the following summer as part of their more formal research program in the summer there. That's just the kind of person that Webb is. He's basically driven and does what he needs to do to succeed. So after his first year in medical school, as if med school wasn't enough, he applied to be in their medical sciences training program, where he's going to earn an MD and a PhD, working under the direction of Drs. Shu and Thompson, focused on citrullination type of protein post-translational modification and its role in amyotrophic lateral sclerosis, which most of you probably know better as ALS. Webb's talk is sponsored by the Salem State Department of Biology and Thermo Fisher Scientific. And so on our Alumni Day, I'm super happy to welcome Webb back to give his talk, which is entitled, "What's the Role of Citrullination in ALS?" Thank you.
All right. Welcome, students, faculty, distinguished guests, to my talk today, which is, as Dr. Brown just mentioned, titled, "What is the Role of Citrullination in ALS?" And just to begin, it is citrullination. Yes. All right. So my talk will be split up into three parts. The first, I will give you some basic definitions and some background so that we all have a fundamental knowledge of what I will be discussing further down the line. Then in the second part, I will be talking about a recent discovery by my lab and what it may mean for future research. And finally, in part three of my talk, I will discuss how I plan on moving this work further, ultimately doing my thesis within this project. And so to start, I want to just give a background on what citrullination actually is. So citrullination is a post-translational modification, which is when proteins are modified after they are translated. And what we're seeing here is phosphorylation, which I'm sure many of you are familiar with. Phosphorylation is similar to citrullination, except citrullination is a conversion of arginine to the amino acid citrulline, and it's catalyzed by a family of calcium-dependent enzymes known as protein arginine deaminases, or also known as PADs. And these PAD enzymes perform this hydrolysis reaction that converts the guanidinium group of the arginine to a urea group within the citrulline. And through this reaction, we get a loss of a positive charge and a loss of 2 hydrogen bond donors. And so we can see that a change from one amino acid to another may cause some deleterious effects within the protein backbone. And citrullination occurs in disease as well as normal physiology. However, in normal physiology, the intracellular calcium levels are just not quite high enough to activate these enzymes. However, there are some instances where this does occur, such as in the skin, where our keratin, which allows our skin to be water resistant, this protein is citrullinated. And citrullination of this protein prevents the cornification or plaque buildup. And some recent studies have suggested that patients with psoriasis lack the citrullination of their keratin, which leads to the plaques that are characteristic of that disease. We also see citrullination in gene regulation, where cells that have increased citrullination of histones, which are proteins that wrap DNA, we see an upregulation of genes that lead these cells to be more stem cell-like or pluripotent. And the opposite is true, where a reduction in citrullination leads to a differentiation of these cells, upregulating genes for tissue determination. And we also see citrullination in apoptosis, where citrullinating vimentin, which is a filament of the cytoskeleton, leads to a disassembly of the cytoskeleton and ultimately leading to apoptosis. And one of the most common citrullination events, which is part of our innate immunity, which many of you probably have seen before, where if you get a cut or some kind of assault to the body, we see pus formation. And in that pus formation is where our neutrophils, which are kind of the first responders to any infection that we have, they citrullate their histones and lead to unraveling of their DNA. And that unraveling of their DNA ultimately bursts the cell, killing it in the process. But through this mechanism, the DNA wraps around pathogens or bacteria and prevents further downstream infection. And so citrullination, having some few instances that we see in normal physiology, majority of the citrullination events that have been studied are found in disease and dysregulation of pads. Specifically in rheumatoid arthritis, where this post-translational modification is a hallmark, where the same mechanisms as a previous slide occur, where these neutrophils infiltrate into the joint space and they burst open, releasing those enzymes with citrullinated proteins within the synovial tissue. And those proteins can then re-enter the body and into the bloodstream, which we use in the hospital to determine if someone has rheumatoid arthritis, using it as a diagnostic biomarker for anti-citrullinated proteins. More recently, there's been some revelations in understanding citrullination within the context of neurodegenerative diseases. And in Parkinson's disease, what we're looking at here is a half slice of the brain, and the green mark here is our midbrain, which sits right between our brain stem and our cerebrum. And in the midbrain, we have a cross section here. We have an area called the substantia nigra, and this is where dopaminergic neurons, or neurons that produce dopamine, reside. And more research has been shown that citrullinated proteins are abundant within the surviving dopaminergic neurons in the substantia nigra in patients with Parkinson's disease. In multiple sclerosis, where citrullination was one of the main mechanisms by which of myelin protein degradation, what we're looking at here is a cross-section of the brain using a CT scan, and the white areas that we are seeing are areas of demyelination. And that is where the myelin, which wraps around our axons, start to degenerate. And what we have seen is that in the myelin basic protein, which is a major protein that makes up myelin, is majority citrullinated. And lastly, in Alzheimer's disease, what we're looking at is another cross-section of the brain. And here we're looking at the hippocampus. And the hippocampus is the part of the brain that encodes long-term potentiation or encodes our memories. And what we have seen recently is in postmortem tissue, so after patients have passed, we've noticed that there is increased protein citrullination within the hippocampi of these patients. And so given these new studies on understanding citrullation within the context of neurodegenerative diseases, my lab decided to focus on ALS. We wanted to determine whether if citrullation is occurring within ALS as it is occurring in other diseases. And so I want to give a quick background on ALS so that we all understand the disease. So ALS, or as we call it, amyotrophic lateral sclerosis, is a neurodegenerative disease that's characterized by selective motor neuron degeneration. ALS is split into two camps, where 90% of cases are sporadic, meaning that there isn't a defined cause through environmental or genetic factors. However, 10% of ALS is familial, meaning that there are defined genetic mutations that are passed through families. And some of these mutations that have been recognized are C9orf72 repeats, which leads to dipeptide repeats. We have SOD1, which is probably the most known and most studied, where this antioxidant protein becomes mutated and ultimately aggregates. And aggregation is typically found in ALS, where all these other genes also perform the same mechanism, where TDP43 or TARD-BP is a DNA RNA binding protein that's known to also aggregate, and also FUS, which is a fusion sarcoma protein, also known to aggregate. And so in ALS, the motor neuron degeneration does occur over time. However, in normal physiology, we see that the motor neurons propagate action potentials that innervates the muscles and ultimately lead to voluntary movement. And that's how we're all able to walk into this room today. However, in ALS, these motor neurons degenerate, and the lack of stimuli to the muscles lead to atrophy, as well as a host of other symptoms like rigidity, hyperreflexia, weakness, and fasciculation. And here we are looking at mice models. One is a wild type and one is ALS. And as the timestamp goes on, we will see and recognize the ALS mouse. And what you can see is that similar to the wild type mouse, you really can't distinguish the ALS mouse from its counterpart. However, as the disease progresses and the mice age, we do see that there's a lack of movement. And that's because of the motor neurons that start to degenerate. And as the mice grow, even as the mice progresses, we see that weakness starts to occur as more and more neurons start to die. However, as the mouse continues on, a lot of those motor neurons do die, and the lack of stimuli to those muscles that I mentioned creates paralysis. And so they can't move any of their limbs. And this is something that we see in patients as well. And so what we saw here was a progression of the disease where pre-symptomatic mice run around like regular mice. And we see this in patients where you wouldn't be able to determine at a young age if someone is going to get ALS. And so they are similar like everyone else. However, as the disease progresses, specifically in the 6th and 7th decade of their life, the motor neurons start to degenerate under unknown mechanisms that leads to weakness and ultimately to a paralysis, where a lot of these patients lose the ability to walk and find themselves immobile. And ultimately, these patients do pass, unfortunately, through the degeneration of motor neurons that propagate action potentials to the diaphragm. And due to the lack of stimuli to the diaphragm, they lose the ability to breathe. And so you can see how important it is for us to delve deep into this research to really understand what is happening in ALS and potentially bring some therapeutic modalities into the clinic. And as these motor neurons degenerate, there's been some mechanisms that have been identified to specifically cause motor neuron degeneration. The first being glutamate excitotoxicity, where excess glutamate released by a presynaptic cell activate the postsynaptic neurons and overcharge the neurons so that to the point that they are overstimulated, and that leads to neurodegeneration. There's also DNA and RNA dysregulation that's also been found to lead to the degeneration of these neurons, as well as some mitochondrial dysfunction, where the mitochondria lacks the ability to output a lot of energy, but seems to also increase its ability to generate reactive oxygen species, which are detrimental to the cell. And one of the main hallmarks of ALS, which is seen in all of ALS, is protein aggregation. And this is where inclusion bodies of various proteins are found within the motor neurons and ultimately leading to a dysregulation of downstream events. And there's also a cargo dysregulation, a dysfunction of the transport down the axons that will ultimately innervate these muscles. And the trapping of these cargoes within the axons can lead to degeneration. And lastly, there's been recent studies that looks at non-cell autonomous mechanisms of neurodegeneration, where glia cells like astrocytes and microglia can lead to neurodegeneration by increasing neuroinflammation. And so that concludes the first part of my talk, and I hope everyone's able to understand ALS a bit better so that we can dive deeper into some of this new research that we were able to uncover within our lab. And so in September, we published a paper titled Protein Citrullination Marks Myelin Protein Aggregation and Disease Progression in Mouse ALS Models. And the two mice models that we use are SOD1 and PFN1. SOD1 is an antioxidant protein that is ubiquitously found throughout the cell. And the mutation that happens is a missense mutation, and that means one amino acid is changed. And what we're seeing this depiction is a glycine at the 93rd position was switched to an alanine. And just that one switch of the amino acid causes ALS. And similarly with the profilin, which is another protein that causes ALS, a similar missense mutation where cysteine at the 71 position is changed to a glycine, and that also leads to ALS. And in both cases, these mutations lead to the proteins to lose its function and ultimately aggregate within the cell, preventing any kind of cellular processes from occurring. And so given this background, I want to give a quick overview of the experimental approaches that we've used so that we all understand what we are looking at. And so we performed Western blot assays to determine protein of interest. And the way we do this is we get a gel and we run proteins on it. And you can get proteins from your blood or from the central nervous system. And when you run this protein, you're able to separate them by size using electricity. And from there, you can transfer these proteins onto a membrane. And after you transfer them, you can probe them with antibodies. The first antibody will recognize the protein that you are looking for. And the second antibody will recognize the first, but it will also have a fluorescent tag that allows us to identify the proteins that we are looking for. And so through this process, if we are looking for a protein at this specific size, we're able to determine that it is present within our samples. And so that is what we saw here, where we are looking at Western blot assays where we ran the spinal cord of these mice on our membranes. And what we saw here is our pre-symptomatic, which are mice before you see any symptoms, our onset, or is when the mice start to become weak, and then paralysis, or when the mice become immobile and are unable to move. And the NTG next to them are non-transgenic, which means these mice do not have any genetic manipulation. So these are the wild type versions. And so what we see here is that there seems to be in both our SOD1 and our PFN1 mice, there's this increase in protein citrullination within the paralysis stage of these mice. And we see that also in the onset. And so this experiments suggest that protein citrullination seems to be a hallmark of the disease, similar to other neurodegenerative diseases that we're able to study. And more specifically, it seems to coincide with disease progression. So as the disease goes from pre-symptomatic to onset to paralysis, there seems to be this increase in protein citrullination. And moving forward, I will be showing data just on the SOD1 mouse, but keep in mind that this data has been seen in both our mice models. And so next, we wanted to answer this question. Now that we know that ALS has this aberrant protein citrullination, where is this citrullination occurring? And so we decided to split the central nervous system into 4 parts. We have the CSC, which is our cervical spinal cord, the BS, which is our brainstem, the CB, the cerebellum, and CTX, which is the cortex, which is the bigger part of our brain. And what we can see is that compared to our wild type or non-transgenic, at least in our CSC or cervical spinal cord and BS brainstem, we see this increase in intensity that is identifying proteins that are citrullinated. And specifically why this is important is because those are areas of the brain that have severe neurodegeneration. So as axons travel down the brain to the spinal cord, these areas have abundance of those motor neuron axons that are degenerating. And so this data, if you look at the quantifications, we can see that it is significantly increased within this area of the brain. And this suggests that citrullination is happening within ALS and that it's happening at the areas in the brain and spinal cord that are severely degenerating. And so the act of motor degeneration is associated with protein citrullination. And so given these new revelations, we wanted to further deep dive and understand what are those proteins that are citrullinated. And to do so, we did a simple experiment where we spun down the spinal cord of these cells. And by doing so, we're able to separate soluble proteins, which will stay in the supernatant, and proteins that are aggregate, which will fall down to the pellets. And again, we have our non-transgenic and pre-symptomatic onset and paralysis mice. And what we saw was that all of the protein citrullination is found within the pellets. And specifically, we see it only in the onset and paralysis stage. And when you look at the quantifications, we can see that it is significantly increased compared to their age-matched transgenic controls. And so this data suggests that citrullinated proteins are found within aggregates in the spinal cord in mice with ALS. And so understanding that the aggregates are the majority of the citrullinated proteins, we wanted to further deep dive into understanding what proteins are actually citrullinated. And so to do so, we performed another experimental approach. This time, it's immunofluorescence microscopy. And in immunofluorescence microscopy, we're able to take our mice, remove their spinal cord and brains. Then we can cut their spinal cord into thin slices so we can put them on slides. And from there, similar to our Western blot, where we can probe them with antibodies, the first being an antibody that recognizes the protein of interest that we were looking for. And the second antibody, as before, recognizes the first and also has a fluorescent tag that allows us to determine if that protein exists in our sample. And using some computational approaches in the microscope, we're able to shine light at a specific wavelength that allows to see the difference of colors between proteins. And so that is what we saw here, where what we are looking at is GFAP is a protein that marks astrocytes. CITP is our citrullinated protein antibody. And the merge are overlaying the two images together, and the inset is an enlargement of the dotted box that you're seeing. And so what we saw here is that in our pre-symptomatic and our onset, in our pre-symptomatic and our non-transgenic mice, we don't see much of an intensity with our GFAP or citrullinated proteins, and we can see that corresponding with our merged images. However, as the disease progresses to onset and ultimate paralysis, we see this increase in intensity of the GFAP, which stands for the astrocytes, and our citrullinated proteins. And when we look at the merged image, they beautifully colocalize together, where we see citrullinated proteins and astrocytes occurring in the same areas. And so through these quantifications, we're able to determine that GFAP, the protein in astrocytes, seems to be increasing as the disease progresses, as well as citrullinated proteins, which seem to also be increasing as the disease progresses. And we saw that in our bulk measurements, and it coincides with the results that we see here within astrocytes. And so given this data, we wanted to dive deeper and answer this question of if this bulk measurement that we see in our spinal cord is the same that we see in astrocytes, what do we see in neurons? And so similar to the previous slide, here is our NUN, which is our marker for our neurons. And CITP, again, our marker for citrullinated proteins. And although there seems to be a high background, we were still able to do some quantifications. And what we can see here is if you look at the inset, you can see that there's this co-localization of our neuron with the citrullinated proteins in our wild type mice. These mice do not have ALS. And we see the same result in our pre-symptomatic mice before any symptoms of ALS arise. However, as the disease progresses to onset and paralysis, that co-localization of our neurons with citrullinated proteins seems to be diminishing over time. And so through this quantification, we're able to determine that these results suggest protein citrullination coincides with neurons in non-transgenic as well as pre-symptomatic mice. However, as the disease progresses and the disease gets worse, there seems to be less and less citrullation within the neurons. And so next we wanted to determine what other proteins may be citrullinated. Given that there were some recent studies of citrullinated proteins within myelin proteins like MBP, we wanted to probe a similar approach. And so what we see here is the immunofluorescence again, However, what we're looking at here is PLP, which is a protein that makes up myelin, and there's multiple proteins that do make up myelin, so we were able to look at a few. And here we can see that in our pre-symptomatic and onset, if we look at our merged image, we don't really see much of a colocalization of this myelin protein with our citrullinated protein marker. However, as the disease progresses to onset, we start to see these inclusions in the white matter that look like aggregates that are co-localized with our citrullinated proteins as well as our myelin protein. And as the disease progresses to paralysis, we do see this abundance of these aggregates that are found within the white matter. And interestingly, they are intensed with the citrullinated protein marker as well as the PLP. And so we wanted to further deep dive and look into this. And so we chose another protein, MBP. And as I mentioned earlier, MBP is a protein that is known to be citrullinated in multiple sclerosis. And what we see here is a similar results as our PLP. We can look at the inset, we can see that there seems to be no colocalization between this protein and our citrullinated proteins. However, as the disease progresses, we start to see these inclusion bodies that are formed in the onset. And as the disease progresses, we see a similar result as our PLP, where all of these aggregate formings are in the white matter and that they are highly intense for citrullinated proteins and MBP. And so this allowed us to determine that at least these two are co-localized with citrullinated proteins. And doing other experiments on other myelin proteins like MAG and MOG, we're able to determine that PLP and MBP are two proteins that have a high co-localization with citrullinated proteins. And so this experiment tells us that the myelin proteins, specifically MBP and PLP, are citrullinated within ALS, and that citrullination seems to be increasing as the disease progresses. And so this paper was revolutionary in terms of bringing to the forefront the idea of citrullination occurring in ALS. And it was able to give us some insights, such as protein citrullation is a hallmark of ALS, particularly in late stage, where we see citrullinated proteins within the spinal cord of the paralysis mice. Citrullination seems to also occur in areas that have severe neurodegeneration. And so citrullination and the motor neuron death seem to be associated with each other. And there also seems to be this increase in protein citrullation overall in these cells, these astrocytes, which we do see in the bulk measurement. However, the opposite is true with our neurons, where citrullination seems to be decreasing as the disease progresses. In the myelin protein aggregates that we see in multiple sclerosis are citrullinated in ALS. And so this experiment, this paper allowed us to determine that citrullinated proteins are a new class of aggregates found in ALS. And there are other aggregates that are found in ALS, and citrullinated proteins now is just one of them. And although this publication was able to really answer this question, this fundamental question of if citrullination is occurring in ALS, it doesn't answer all of the questions. And so there's a lot of questions that are still unknown, like what other proteins are citrullinated? It would be interesting to know why certain proteins are citrullinated and others are not. Also, does citrullination cause protein aggregation? After seeing those protein aggregates within the white matter, it's understandably that we want to understand if citrullination may be causing those proteins to become less soluble and ultimately become inclusion bodies within the cell. And also, does citrullination cause ALS, or is it a consequence of the disease progression? So we don't yet know if when citrullination occurs, if that is the trigger that leads to ALS, or if ALS, as the disease progresses, leads to this increase in protein citrullation. And we also want to answer why there seems to be this difference in citrullination patterns between these cell types, neurons and astrocytes. Why do astrocytes have increased protein citrullination while neurons have decreased protein citrullination? And lastly, what is the consequence of protein citrullation? Is it good for ALS? Is it bad for ALS? And those are questions that we still have yet to answer. And so for my thesis, I plan to answer this last question. And so this brings us to the last part of my talk, where I will be talking about how I plan on moving this work forward. We performed some recent high throughput screenings to identify proteins that were citrullinated. And using our paralysis mice compared to our wild type, we identified many proteins that seem to have an increase in protein citrullation. We see GFAP, the protein that we just talked about in astrocytes. We see vimentin, a protein that I mentioned earlier in apoptosis that's known to be citrullinated. We see albu, which is albumin, a protein in our blood. And so I wanted to focus on this protein here known as HSPB1 or heat shock protein beta 1. And HSPB1 is a small heat shock protein that acts as a molecular chaperone. And so it prevents aggregation of denatured proteins. And you can see how this is important in ALS as the mechanisms of motor neuron degeneration is caused in part by protein aggregation. And looking at the expression of this protein within the body and tissues, the yellow is referring to our central nervous system, and we can see that HSPB1 has a moderate increase in expression within our central nervous system and also in our muscles. And so this tells me that this protein is very important and shouldn't lose any function if it has such a high expression within the central nervous system. And so I chose HSPB1 because it has a role in RNA translation, and it also has a role in cytoskeleton stability. It also plays a role in apoptosis as well as autophagy when proteins are broken down within the cell to remove unfunctioning protein and ultimately create new ones. But what really drew me to HSPB1 was this propensity to prevent protein aggregation. And as I had mentioned earlier, that there are other mutations that can cause aggregations within ALS, like TDP43, SOD1, and FUS. And surprisingly, all three of those proteins are regulated by HSPB1. And so my hypothesis is that citrullination alters the chaperone activity of HSPB1. And so to investigate this hypothesis, I've proposed these two aims. The first being to investigate how citrullination affects the chaperoning function, and the latter being how citrullination affects the TDP43 interaction. And so to study the first aim, I will take the human protein, HSPB1, and incubate it with the PAD2, which is an isozyme of the PAD enzymes that is predominant in the central nervous system and leads to the downstream effects of protein citrullation. And I will incubate it in the presence of calcium, given that these are calcium-dependent enzymes. And from there, this protein will be citrullinated. And then I will take the citrullinated form of HSPB1 and incubate it with citric synthase. Many of you probably know citric synthase as a protein in the TCA cycle or the Krebs cycle that plays a role in respiration. And there's been studies that shows that HSPB1 as a chaperone protein is able to prevent citric synthase from denaturing. And when you add heat to the system, citric synthase tends to break down. And once you remove that heat, the breakdown of that protein tends to be reorganized by HSPB1. And through these experimental approaches, I want to understand how citrullination of HSPB1, how does it affect the chaperoning function and prevent citric synthase from remaining as an aggregate using our aggregation assays. And I won't go too in depth on these assays, but it essentially tells us that is citric synthase an aggregate or is it soluble? And next, I want to understand whether citric synthase regains its function. And as I mentioned, it is a protein within the TCA cycle. So it plays a pivotal role in respiration. So I want to understand, after this protein is degenerating, does HSPB1, through its chaperone function, able to reorganize this protein so that it regains its function? And so this assay will tell me whether citrullination prevents this from occurring and ultimately leading to citric synthase losing its activity due to being an aggregate. And so these experiments here will tell me whether or not that citrullination of HSPB1 affects the chaperoning function of HSPB1. And so our next approach will be understanding how it affects the interaction of TDP43. There's been recent revelations that shows that HSPB1 directly binds to TDP43. And as I mentioned, TDP43 is a protein that aggregates in ALS. And TDP43 pathology, where the wild type protein, so the protein that doesn't have any mutations, the protein that you have in you at this moment, seems to aggregate as the disease progresses in 90% of ALS, whether it's sporadic or familial. And so I thought the interaction between HSPB1 and TDP43 seems to be an appropriate way to understand how citrullination may be affecting this interaction and ultimately leading to aggregation of TDP43. And so through these experiments, I want to incubate our HSPB1 in red and TDP43 in green in these in vitro systems, so in a tube, cell-free. And from there, I want to understand whether TDP43 co-localizes with HSPB1. And when TDP43 aggregates, it goes through a phase transition. I'm sure many of you know what that is. When ice turns to water and ultimately to steam, that is a phase transition. And the opposite is true for TDP43, where it is in a soluble liquid phase and ultimately transitions into solids, and that is what causes these aggregation. And we have seen that HSPB1, the wild type version, prevents this aggregation from occurring. And through these experiments, citrullinating HSPB1, we want to answer this question, whether the citrullinated version of HSPB1 is still able to prevent TDP43 from transitioning from its soluble phase to a solid. And then next, I want to perform the FRAP assay, which is a fluorescence recovery after photobleaching, where we take these droplets of TDP43, which are liquid in nature, and we bleach areas using photons. And from there, the liquid property of the droplets should allow the free flowing of the molecules so that it covers up the area that has been photobleached. So over time, we should see a normal droplet if it is still liquid and it's moving in a non-rigid way. However, what we think is happening is when HSPB1 is citrullinated, it prevents the liquid form of TDP43 and ultimately leads to the aggregation of TDP43. And so if we're able to photobleach an area of the droplets, if these droplets have been turned to aggregates, there's going to be a lack of the free flow of those molecules. And so that area that we photobleach will still be visible seconds and minutes after. And lastly, it will help us, using a filter trap assay, will help us really determine what we see here, if it's true, using the filter trap assay. And this filter trap assay is a very simple experiment where we run our samples through a filter, and the filter has very small pores and prevents aggregates from flowing through. And these aggregates are trapped on the membrane, and we use it like a Western blot where we can determine if those protein aggregates are present within the samples. And so what we expect to see is that with TDP43 and HSPB1, the wild type version, TDP43 remains liquid. And so it should be able to flow right through these filters and not be on our membrane. However, when HSPB1 is citrullinated, we expect the interaction with TDP43 to be compromised. And so TDP43 will ultimately form these large aggregates that will be stuck on the membrane and will prevent it from flowing through. And using these antibodies, we're able to determine if TDP43 is bound on our membrane as an aggregate. And so these experiments here will illuminate the role of citrullination in the context of HSPB1 and better understand how citrullination affects proteins and their function. And in the case of HSPB1, there are multiple outcomes. The first being, it is possible that citrullination completely removes the chaperoning function of HSPB1. And as I mentioned, it is a protein that organizes lots of proteins that aggregate in ALS. And so we can see that if citrullination of HSPB1 does compromise its chaperone function, that it could lead to downstream effects of multiple proteins aggregating, ultimately leading to motor neuron degeneration. Another outcome is that citrullination actually increases the chaperoning function, although we think this is less likely given some of the previous experiments that we have done. And in this case, citrullination of HSPB1 would ultimately lead to many proteins remaining soluble within the cell. And lastly, it's also possible that citrullination does not affect the chaperone function of HSPB1. Wild-type HSPB1 has a functioning chaperone function, which means if HSPB1 is citrullinated and that is not disrupted, then it will act in its normal way. However, out of these three, we anticipate that the most former is the most likely case, where citrullination of HSPB1 is really going to hinder its ability to prevent protein aggregation within these cells, ultimately leading to large aggregates that kill these motor neurons and ultimately lead to paralysis that we see in ALS. And so this experiment here will give us more insight into why these protein aggregates seem to be forming within ALS and ultimately causing these motor neurons to degenerate. And so today I've talked to you about citrullination and its role in multiple neurodegenerative diseases. And more recently, our lab has been able to identify citrullination as a hallmark of ALS. And more specifically, there are specific proteins that seem to have an increase in citrullination. And through these experimental approaches, we plan to understand what is the consequence of this protein citrullination and whether or not it is contributing to this mechanisms of ALS. And so these experiments, along with others, will give us an idea of how we should move forward within this field, and ultimately would bring new avenues of investigation so that we can better understand how other proteins are affected by citrullination. And given that these PAD enzymes are dysregulated in causing these aberrant protein citrullination, it potentially could be therapeutic if we determine that citrullination is bad for ALS. And there's been some recent studies that shows the efficacy of drugs that inhibit this protein. And lastly, the most promising is that these citrullinated proteins seem to be coinciding in specific disease progression. And as we saw that there seems to be citrullinated proteins within the onset as well as the paralysis state, we can then identify proteins that are citrullinated at a given stage. And from there, we could use those proteins as a diagnostic biomarker to determine if someone has ALS. And this would be significant because ALS is a diagnosis of exclusion. When you are determined to have ALS, it's after they have done extensive tests to rule out every other possibility. And that is the end of my talk. And I want to acknowledge my labs, the Shu lab and the Thompson lab with Dr. Thompson and Dr. Shu, as well as all of our lab members. I want to thank the NIH for funding, as well as the ALS Angel Fund. I also want to thank the UMass MD, PhD program for all of their help and support, as well as the School of Medicine, where all of my colleagues will be graduating this year, and the Graduate School of Biomedical Sciences. And I also wanted to thank my nonprofits, One Leaf, as well as Salem State, for allowing me to come and speak to you all today. And from here, I can take any questions. There's a question on that. There's a question over there. Yeah. Yes.
Thank you for this talk. I have a question regarding the therapeutic approach. As far as I understand, how the two look with similar process and relation has been seen connects to Alzheimer's disease. Is there any therapeutics to control the relation? I also understand that it doesn't modify once it happens, you get it first, right? So can you control the levels of citrullination and medical approach?
Yeah, so the question is if there are any therapeutics that can control citrullination within the context of other neurodegenerative diseases such as Alzheimer's disease. And unfortunately, not in clinic. However, we do have some drug targets that are able to prevent citrullination from occurring on a basal level. So we have drugs from our lab that has been shown to prevent all of the isozymes of PADs. So PADs have 6 isozymes, kind of a misnomer. There's only 5, but it's 1, 2, 3, 4, 6. And all of them are able to be inhibited by these drugs that we have developed. And we've also developed some PAD specific, like PAD2, as I mentioned, was the isozyme specifically in the central nervous system. We have developed drugs that specifically target PAD2 and PAD4. However, some of these drugs are still in trial and we want to understand their toxicity before we bring them to the clinic. But hopefully in time, we're able to dive deeper into understanding how citrullination affects these diseases and whether or not these therapeutics would be a great approach for that.
Aging talk that has taught me a lot more about ALS. I'm responsible for connecting you to our online audience that numbers over 130. And the first question I have is from one of our students related to your work at the Chan School of Medicine. And I'll read the question if you could also repeat it. What groups of people work in your ALS lab? Are there, in addition to your colleagues, interns, undergraduates, etc.?
So unfortunately, UMass Medical School is just a graduate school, so we do not have any undergraduate students. However, in our labs, we are welcoming to any students who want to dive deeper into these research. And we take research techs. If anyone is graduating and is looking for a job, we are looking for research technicians and other positions within our lab. And so you would be working with me and really diving deep into these experiments.
Before I ask the next question, which is actually from the same student, please remember everyone that in 25 minutes, whether you are here in this room or watching from your dorm, there's going to be a lot of pizza that Webb and I will not be able to eat just between the two of us. So do come and join us and you'll be able to chat with Webb over a pizza lunch. Webb, the second question, which was from the same student, I sent him a link to the summer research program that the Chan School of Medicine has. I know you're not necessarily involved with that program, but could you tell us a little bit about that program?
Yeah, so the question is to give an overview of the summer research program at UMass Medical School, which I had the opportunity of participating in. This program is sponsored by NIH, and they accept about 25 students every year. And you spend 10 weeks in the summer living in Worcester and doing research at UMass. And you get to choose which labs you want to do research in, and you do get paid quite a bit of money, which is also a good thing. But it gives you an opportunity to get your hands dirty and understanding how experimental approaches are done and how we think about science and bringing the scientific method to work for the questions that we are asking.
There are more questions in the online audience, but I don't want to dominate. Are there any in the?
Thank you.
Here's the point, if you know if there's any connection between the protein aggregation and defects in axon transport that you mentioned.
Yeah, so there seems to be aggregation of multiple other proteins, and when proteins start to aggregate, they can actually trap wild type proteins within those aggregates. And sometimes what happens is as cargo is being transported down the axons, these aggregates can become so large that they're trapped within the axon. And as you can see, if that entrapment does occur, a lot of cargo cannot continue down the axon. And so the axon loses a lot of proteins, specifically like cytoskeleton proteins that maintain the axons, and ultimately leads to like Wallerian degeneration, where the axons start to die at the end and the degeneration moves forward towards the cell body. And so we don't yet know how citrullination plays a major role in that. However, multiple proteins aggregating and causing these cargo dysfunction is well documented within ALS. Mhm. Yeah, so tubulin luckily is not affected, or I would have to find a different loading. But other proteins, cytoskeleton proteins, are citrullinated. However, in our high throughput screening, we didn't identify high levels of citrullination. And even in our wild type mice, we do see some level of citrullination occurring at the cytoskeleton proteins. Oh, sorry, forgot.
This is going to be a lot less sophisticated, right? So you showed us imagery from your immunofluorescence spectroscopy and mentioned that citrullation occurs a lot more in astrocytes. Can you clarify what astrocytes are?
Yeah, okay. So astrocytes are, sorry, the question is, what are astrocytes? So astrocytes are a type of neuronal cell. So they are a subset of the neural progenitor cells, which are cells that also make neurons. And astrocytes play a pivotal role in neuronal homeostasis, in that astrocytes also create the blood-brain barrier. And so it prevents any kind of proteins that shouldn't be within the central nervous system from crossing the blood-brain barrier into the central nervous system. And astrocytes play a pivotal role in energy formation. And so astrocytes can make glutamate as well as other metabolites that are shuttled to the neurons and allow the neurons to have lots of energy, because the neurons are firing at rapid rates. And so the energy that they produce themselves is just not sufficient. And so the astrocytes play an important role in making sure that the neurons have enough fuel and that there aren't any things that are coming into the central nervous system that shouldn't be.
Hi, thank you. Sort of a follow-up to we can back around this question about the therapy, right? So my understanding of these bacteriologists is that you know, there are sort of people when they use neurons, if neurons don't reproduce very much, right?
Right.
Do astrocytes reproduce a lot?
So yes, astrocytes can divide.
Okay,
Yes.
so the follow-up is, you showed some of say that different brain sections
Mhm.
And then it seemed like the citrullinated proteins in normal cells started out in the cell body.
Mhm.
And then as the disease progressed, they moved out into the axon.
Mhm.
Right? So, and you said that there were therapies about inhibiting the patterns.
Mhm.
So do you have any data that shows the migration, like is it just the patterns on these the cell body? And so sort of activated to be there this the marker of degeneration. That's what's going on and that's why you're looking at different patterns.
Yeah, so the question is the mechanisms of protein citrullination, if that's occurring by PAD enzymes being released by cells or in other ways. And so what we have seen is that some cells are different, like there's neutrophils that can cross the blood-brain barrier and they can have the PAD enzymes on their cell surface. And so you can imagine that in the extracellular space, there is enough calcium levels to activate those enzymes. And if there are proteins within that area, it can citrullate them. However, we don't fully understand what is happening within astrocytes because we don't fully yet know if these proteins are being released by astrocytes or if these proteins are being citrullinated and then released by astrocytes. And so this is actually ongoing investigation. And we see this in human tissues where these aggregates are forming in areas of the white matter where astrocytes typically do not reside, and these astrocytes seem to migrate to those areas, and we're not sure yet if they're causing the protein citrullation from internally and kind of spilling that out, or if those astrocytes are also dying and ultimately releasing their proteins and causing protein citrullation. So I think in all of this aspect, it would be good to have therapeutics because they don't necessarily work only intracellularly. So if we have therapeutics that cross the blood-brain barrier, that therapeutic molecule can be within the central nervous system through the spinal cord and the cerebral spinal fluid. And so it can be around the extracellular space as well intracellularly. So these therapeutics in theory would be able to prevent these protein citrullation from occurring either intracellularly, if that is what is occurring, or extracellularly, which we see these aggregates forming.
Webb, thanks again. I have a few more online questions. We'll take 2 more online questions, and I've been told that you don't have to repeat my questions to you because I'm talking to the online audience as we speak. And very quickly, just speaking to the online audience, your attendance is taken automatically if you're logged into this webinar. So my question to you, Webb, the work you're doing on citrullination on ALS, will it ultimately lead to more efficient or effective treatments and maybe even cure ALS?
Here is a high bar, but that's what we aim for in science, right? I do think that this work does have some high implications in terms of some therapeutic modalities that can be ultimately used within the clinic. If we're able to determine that protein citrullation is bad for the disease and it is happening after the disease is occurring, it is possible that we could reduce the symptomology of the disease by reducing citrullation. And given that these proteins are aggregating, if we're able to determine that these aggregations are in part associated with the protein citrullation, if we have them therapeutics that can prevent protein citrullation, in theory, we could prevent or reduce the aggregation, ultimately reducing the motor neuron death. And so these patients would be able to live much longer without having any kind of immobility problems.
And then a final online question, and just to remind our online audience, if you're within 10 minutes of this room, Betts Hall Campus Center, you can come and physically ask Webb the question over pizza. The final question, Webb, is what drew you to research on ALS itself?
So I initially, coming at Salem State, my dream was to cure Alzheimer's disease. I had some very high ambitions while I was here as a student. And when I had a chance to rotate within labs, for me, what drew me to ALS was mainly my mentor. My mentor is also an MD PhD and felt that connection with him. And given some of his track records and identifying some novel occurrences, novel mechanisms that are happening within ALS, I felt that this research could be very pivotal in understanding how protein citrullination may be affecting not only ALS, although this work is in ALS, but this citrullination is happening in all other neurodegenerative diseases like Alzheimer's and Parkinson's. And so this work could potentially be moved to these other diseases and ultimately lead to therapeutics that could help all neurodegenerative diseases. And as the population ages, we do see that neurodegenerative diseases will be more and more prevalent as time goes on. So it would be very imperative for us to put a lot of effort into understanding how these neurodegenerative diseases can be mitigated.